started out 7-8 years back. Our mission was to disrupt the cost barrier for biosimilars, and to do that, we were looking at disruptive technologies that will allow us to significantly reduce the cost of manufacturing. And when you look at fed batch, the biggest footprint guzzler in a sense are all the whole tanks. So when we started doing this, we said, hey, if we want to have a compact manufacturing and impact cost of manufacturing through having a smaller footprint, we should get rid of all of the whole tanks. And that's how we came up with a continuous manufacturing process, which is end-to-end continuous. And the advantage of that, of course, is that you have a very small footprint. So the first version of this is called the EnzeneX platform. And we've launched multiple biosimilars, so it's a commercialized platform. We have 3 molecules commercialized with it in India, and we've done a lot of early development for clinical manufacturing for novel molecules as well. There are different ways to look at continuous manufacturing. Right? So some people, what they do is they run multiple fed batch bioreactors in series with a single downstream. So you run a campaign mode and they call it continuous, but that's really taking fed batch and running it simultaneously. Then there are folks who do perfusion, and then every day they'll take a harvest and do the downstream processes, right? So for these two, I see them more as a conventional manufacturing. You're just managing the manufacturing campaign, right? Then there are folks. There is another company that does protein A capture as a first step, right? But ours is a truly fully connected continuous platform where we start with perfusion, capture it on protein A, followed by pH inactivation, followed by chrome 1 and chrome 2. So all of these steps happen in a fully continuous mode. And so it's truly a end-to-end continuous platform, perhaps the only end-to-end continuous manufacturing platform that is being offered by a CDMO right now. Generally, right now, we are getting around 10 kilograms per month from a 500 liter bioreactor, right? For comparative fed batch clones, which can give around 2 to 3 grams per liter, okay, just as a reference. So the cost of goods savings can be around 40 to 50%. For some more challenging molecules for fed batch, they can be even bigger. Generally, we get 40 to 50% savings in cost of goods, and we feel that there is going to be a significant savings on carbon footprint as well, because we have a very small footprint. So best case scenarios right now, as I was saying, for difficult to express, of course, it's a no-brainer. Where fed batch processes do not work, you have to go to perfusion and continuous manufacturing. So that's a no-brainer. But even for monoclonal antibodies, we are slowly reaching that 40 kilogram per batch, which will allow us to compete with the higher scales of 10,000, 15,000 for fed batch. So EnzeneX 2.0, our target is to take it up to 40 kilogram per batch. We'll be scaling up from 500 to 1 KL. We have some PAT that we'll do. And we have clones that have a higher titer to start out with, right? So when you go at 40 kilogram scale, you can essentially use this for pretty much any of the applications that are currently being used with fed batch and still get the same kind of cost savings. This platform can be applied to pretty much all protein modalities that are out there. MABs are naturally work with it because of protein A, but we've done some non-MAB molecules for early stage discovery companies. We are setting up in the US with the goal of really being partners for early stage companies that do not have a local CDMO in the US. So naturally, that's our ideal partner in a sense. But we are also collaborating with big pharma, and as I was saying, this technology can be useful for pretty much all biotech companies that are out there. We are going to be in the Princeton West campus, and for the first stage, we are going to have our continuous manufacturing platform that is, very well commercialized in India. We want to get that platform here because we have a lot of partners for whom we are already doing phase 1 manufacturing with EnzeneX. So the first lines that we are going to have are going to host the EnzeneX platform. But we are looking at perhaps also going to more automation, as well as having PD lab to support early stage work, right? So, in our phase 1, we are going to host the EnzeneX platform along with process development capabilities that will support tech transfer as well as early development for our US based partners.